Hello everyone and welcome to Burn Your Books, Open Your Mind. I am your friend and host BYBOYM and you are watching Foundation for NEET. We have already started with the first chapter in our course, that is Life Processes, Digestion and Absorption. Today we are beginning with the sixth topic, Photosynthesis Experiments and Activities. We have already understood in our last lecture that green parts of the plant such as the leaf contain mesophyll cells that in turn contains chloroplasts. What are chloroplasts? Chloroplasts are the organelles in which chlorophyll is present. Chlorophyll is the pigment that absorbs light, and this is how the green parts of the plant synthesize food, that is glucose, which is stored as starch. So we know that for photosynthesis light is required, carbon dioxide is required and water is required. So today we are going to start with a few activities that would help us understand how science happens. So let's begin. The first activity is to determine whether chlorophyll is required for photosynthesis. So we are today going to find out whether or not chlorophyll is required for photosynthesis. How do we do that? We take a plant with variegated leaves. Variegated leaves means a leaf that has both green as well as non-green regions. So the green regions are look green because of the presence of chlorophyll. So what would we do next? We will keep this plant in dark for 3 days. Sir, what would happen then? Keeping the plant in dark for 3 days would make the plant use up all the stored carbohydrates. What did we talk about in the last lecture? We said that the plants store the glucose formed in photosynthesis as starch. Starch is a storage polysaccharide. In contrast, in humans, glycogen is a storage polysaccharide. So the plant unable to synthesize food in the absence of light will utilize all the starch that was present. So we'll have a plant that has no more starch in it. What would happen next? We will then keep this plant in sunlight for a few days. And what would happen then? Keeping the plant in sunlight again would make the plant do photosynthesis again, making its own glucose and starch again. We then pluck a leaf of this plant and mark the green and non-green regions. We then keep this leaf inside boiling water. What would the boiling water do? The boiling water would, first of all, it would kill the leaf, it would disrupt the cell membrane and soften the cell wall and cuticle. Okay, after this we keep the leaf in alcohol. Because we cannot keep the alcohol directly in front of the flame, so we use a water bath, a buffer in between. So we again heat the alcohol till boiling. What would this do? What would alcohol do? Alcohol will separate the chlorophyll in the leaf. A dilute solution of iodine is then poured over the leaf and the color change is observed. We would observe that the green parts of the plant that contained chlorophyll would turn bluish black, indicating that starch is present there because iodine links with molecules of amylose that are present in starch. So only the part where starch is synthesized would turn into bluish black. So we observe that the non-green parts of the plant do not change color to bluish black, but while the green parts do due to the presence of starch. The conclusion is that starch is produced only in those parts of the plant where chlorophyll is present. Now moving on to the second activity. Now the aim is to determine whether carbon dioxide is required for photosynthesis. The first was to determine whether chlorophyll was required. Now we are making sure whether carbon dioxide is required. So the procedure is to take two plants and we keep them in a dark room. Again, what would happen? All the starch in the plant would be used up. The plants are then placed in a special setup. We take the plant A and we put the glass watch containing potassium hydroxide along with a bell jar over it. So we have a setup where we have a plant A and a watch glass containing potassium hydroxide and a bell jar over this setup. This is our setup A. In setup B we have a plant B and just a bell jar over it. Plants are then covered with glass jars and their bottom are sealed with Vaseline to make sure that this is airtight. So this is our setup A and setup B. We then place the plants in sunlight for several hours. So we kept them in dark, no photosynthesis, no starch left. Now we place them in sunlight, so again photosynthesis would happen. So we then pluck a leaf from each plant and then we will test it. So from setup A and setup B, what would we observe? We observe that the plant in setup A do not show bluish black color. Why? Because no starch is synthesized. Why? Because all the carbon dioxide was absorbed by potassium hydroxide. So no carbon dioxide was available for photosynthesis for plant A. So no photosynthesis in plant A means no bluish black color in setup A. While in setup B, since the plant was able to photosynthesize as there was no potassium hydroxide to absorb the carbon dioxide, there was photosynthesis in the case of plant B, and the setup B shows bluish black color. So we conclude that setup A has no carbon dioxide, but setup B had carbon dioxide, and carbon dioxide is necessary for photosynthesis. I hope you enjoyed the video. You are watching Burn Your Books, Open Your Mind.